Welcome to Parkinson's disease, the master class. This lecture will introduce the subject of non-invasive stimulation of deep brain targets and provide a glimpse of novel treatments for these brain structures. Presenting the information for this lecture is Dr. George W. Cochran. Dr. Cochran is a chiropractic neurologist with over 20 years' experience in treating complex neurological conditions. Without further delay, here is Dr. Cochran.
This slide will examine the neurological targets for deep brain stimulation in Parkinson's disease. We'll also touch on the potential of non-invasive methods to activate these same deep brain targets. Please direct your attention to the illustration on the right side of the slide. You'll see the subthalamic nucleus highlighted in light green, the pedunculopontine nucleus in red, and the globus pallidus in turn is in blue. These three areas are the primary targets for deep brain stimulation, as indicated by Mayo and Wang at the bottom of the slide. So what exactly is a non-invasive deep brain stimulus interface? Well, let's start with the definition of interface. It's the place where independent and often unrelated systems meet and interact or communicate with each other. Think of it as a man-machine interface, for example. Now, if we take a look at some of the traditional deep brain targets, like the PPN, we'll find that there's an interface through the vestibular system that may allow us to modulate the activity of the PPN in patients with Parkinson's disease. Similarly, the GPI seems to be activated by ballistic movement. So maybe in their person, the stent can be activated by transspinal direct stimulation and or cranial nerve 5 stimulation. The VIM, another deep brain target, can also be activated by certain types of vestibular stimulation. So here's the idea. We're going to explore the concept of using other systems, particularly the vestibular system, as a non-invasive interface to target some of these deep brain areas. It's an exciting approach that could potentially revolutionize the field. We will delve into the fascinating realm of the pedunculopontine nucleus and its intriguing connection with vestibular input. Recent studies have shown promising results, indicating that stimulating the pedunculopontine nucleus could potentially enhance function in individuals with Parkinson's disease. The ultimate objective is to discover non-invasive methods of stimulating the pedunculopontine nucleus, with a particular focus on utilizing vestibular stimulation. To substantiate this approach, we will examine and analyze existing studies in this field. So let's embark on this journey together. This is a meta-analysis of the PPN. Deep brain stimulation of the PPN has shown significant improvement in postural instability and gait dysfunction, as well as freezing of gait and falling in patients with Parkinson's disease. It is worth noting that patient age, disease duration, medication, equivalent dosage, and the choice of unilateral or bilateral stimulation were similar across all groups of patients with PD. The study provides strong evidence that PPN deep brain stimulation can effectively improve postural instability and gait dysfunction in patients with Parkinson's disease. The findings were published in World Neurosurgery in 2017, making non-invasive neuromodulation of the PPN an exciting and attractive option to potentially alleviate our patients' symptoms. In the next several slides, we'll talk about vestibular stimulation as an interface to this deep brain structure. In the realm of basic science, two significant research papers illuminate the topic of neural circuits and their role in integrating sensory information during movement. These studies examine connections between the vestibular system and the pedunculopontine nucleus, revealing their vital role in coordinating movement and maintaining balance. The first study neural circuits underlying the integration of sensory information during locomotion by Takakatsuki in 2017 provides valuable insights into how sensory signals from the vestibular system are processed by the Pedunculopontine nucleus, fine-tuning motor output, and ensuring stability. In contrast, Peterson's 2018 review paper, Vestibular-Pontine Circuits and the Control of Posture and Locomotion, explores the anatomical and functional connections between these systems, emphasizing their importance in regulating posture during movement. The review also discusses how disruptions in these circuits can lead to disturbances and balance problems. By revealing the complex connections between the vestibular system and the pedunculopontine nucleus, these papers open doors for further research and potential treatments for individuals with gait and balance issues. In human studies, there are two important pieces of research shedding light on the role of the vestibular system in both motor and cognitive functions. One study by Accor in 2016 reviews evidence from human studies showing the link between vestibular problems and difficulties in motor and cognitive skills. It also explores ways to use vestibular system therapies to reduce motor function and abilities in clinical cases. Another valuable study by Ross in 2016 conducted a systematic review on how vestibular stimulation techniques like galvanic vestibular stimulation affect posture control in people with balance issues. This review highlights the potential of using vestibular interventions in rehabilitation programs targeting the pedunculopontine nucleus. In clinical trials, Ferrexi in 2019 ran a randomized trial examining how transcranial direct current stimulation impacts dual task performance in Parkinson's patients. This study focused on stimulating the pedunculopontine nucleus and showed that non-invasive brain stimulation techniques can help regulate pedunculopontine nucleus activity and improve motor skills. Lastly, in 2010, Al-Solahin provided a thorough review on vestibular rehabilitation therapy, outlining its principles and effectiveness in enhancing balance and walking in patients with vestibular disorders. The review stresses the importance of targeting the vestibular and pedunculopontine pathway in rehabilitation efforts. The pedunculopontine nucleus is a crucial part of brainstem associated with Parkinson's disease, particularly in terms of walking and balance issues. When the pedunculopontine nucleus and its connections with brain regions like the basal ganglia malfunction, it can cause motor symptoms such as freezing of gait and instability in Parkinson's patients. K. Ritchie in 2010. Recent research by Perez-Loret in 2012 suggests that vestibular input, which affects our sense of balance, significantly regulates pedunculopontine nucleus activity. This, in turn, influences gait and postural control in Parkinson's disease, dysfunction in integrating vestibular signals within the pedunculopontine nucleus. Circuitry may contribute to the development of motor symptoms in Parkinson's disease. Therapeutic implications and future directions. In the realm of Parkinson's disease, there is great potential in targeting vestibular input and PPN function as a means to alleviate gait and postural disturbances. To fully explore the efficacy and safety of such interventions, further research, including clinical trials, is necessary. This will specifically focus on the vestibular system in Tappan-Free 2010. This summary emphasizes the intricate relationship between vestibular dysfunction, the PPN, and Parkinson's disease. It highlights the importance of investigating these aspects in order to develop innovative therapeutic approaches. Some examples of these approaches include galvanic vestibular stimulation, vestibular skull vibration, and caloric vestibular stimulation. These methods serve as non-invasive alternatives to deep brain stimulation, offering new possibilities for treatment. now let's dive into the VIM nucleus in the thalamus. Just like the pedunculopontine nucleus, it has the ability to interact with vestibular inputs, thus providing a non-invasive method to potentially regulate VIM activity for patients with Parkinson's disease. Numerous studies have demonstrated that direct stimulation of the VIM can effectively alleviate tremors in individuals with Parkinson's disease and essential tremor. Additionally, we will explore other applications and findings that have emerged from research on VIM stimulation. So today we're going to dive into an intriguing brain area known as the ventral intermediate nucleus of the thalamus. What's fascinating about this region is that it can potentially be stimulated without the need for surgery. We'll take a look at the slide, and you'll notice the VIM marked with red double asterisks indicating its location in the thalamus. But here's where it gets even more interesting. There's a connection between the VIM and another brain region called the medial nucleus of the vestibular system. And guess what? This connection holds great potential for using the vestibular interface to influence the VIM and improve symptoms in Parkinson's patients in a groundbreaking study conducted in 2016 and published in the esteemed Journal of Neuroscience. Therapy, researchers delved into how the response to deep brain stimulation in individuals with Parkinson's disease. They compared patients experiencing tremors to those dealing with stiffness and slowness or bradykinesia, and what did they discover? Well, the findings revealed that stimulating the VIM had a more significant impact on reducing tremors compared to relieving stiffness and slowness. This exciting revelation suggests that the VIM could be a promising target for treating tremors in both Parkinson's and essential tremor patients. Now let's delve even deeper into the research surrounding this captivating topic. In a study conducted by Choi and published in the journal Tremor and Other Hyperkinetic Movement Disorders in 2022, the main focus was to examine the impact of deep brain stimulation, deep CIS, on the ventral intermediate nucleus C in reducing upper extremity tremors. However, the study also aimed to explore the potential of VIM stimulation in improving balance among these patients. The results show that activating the stimulator and delivering current to the VIM nucleus not only reduced the severity of tremors but also improved gait and posture. Interestingly, the study revealed additional benefits beyond the expected alleviation of tremors that highlighted improvements in gait, posture, and balance. The researchers concluded that deep brain stimulation of the VIM effectively enhanced gait and balance in patients with essential tremor. The observed improvements in gait, posture, and balance were accompanied by simultaneous reduction in axial tremor during various tasks. This investigation emphasizes the multifaceted role of the VIM nucleus. It not only serves as a target for mitigating tremors, especially in the upper extremities, but also has the potential to influence axial spine function. These findings suggest that modulating the VIM nucleus may have a positive effect on posture and balance control in individuals with essential tremor. In a study published in Brain Structure and Function, Bayer and colleagues 2015 discovered the importance of the nucleus ventrilis intermedius within the thalamus in our perception of gravity. Their research focused on the VIM nucleus and its role in the vestibular system, which was demonstrated through various animal studies. To delve deeper into its function in humans, the researchers conducted experiments involving patients with deep brain electrodes implanted in the VIM area. These patients underwent assessments to measure their subjective visual vertical. The results were fascinating. When the stimulator was activated, there was a slight tilt of 1.4 degrees away from the site of VIM stimulation. However, when the stimulator was deactivated, this deviation from verticality increased significantly to 4.4 degrees. These findings suggest that the stimulation of the VIM region directly influences our perception of gravity and verticality, which is dominated by the otoliths. Overall, This study highlights the close association between the VIM and our perception of verticality. From a clinical standpoint, stimulating the VIM area could potentially shift the center of gravity towards the stimulated side. For example, stimulating the right VIM area might pull the center of gravity towards the right, while stimulating the left VIM area could have the opposite effect, pulling the center of gravity towards the left. This newfound understanding of the VIM's role in modulating our perception of gravity could have practical applications in clinical settings. In a fascinating study published in the Journal of Neurophysiology back in 1978, researchers delved into the world of neurosurgery to explore the effects of stimulating different regions of the brain in human subjects. What they discovered was truly mind-boggling. The patients consistently reported a sensation of moving through space. This mind-blowing experience was linked to a neural pathway called the Vestithalamic Projection, which ultimately connects to a specific area in the thalamus known as the ventral intermediate nucleus. Interestingly, the researchers also noted that the VIM in humans bears a striking resemblance to the Ventral nucleus found in our primate cousins. These groundbreaking findings suggest that optokinetic stimulation, which involves visual motion, could potentially activate the VIM and induce sensations of movement. This opens up exciting possibilities for clinical applications, as optokinetic field stimulation could be used to target and selectively activate the VIM in the thalamus. And the surgeons involved in the study concluded that the induction, of a sense of motion in conscious patients through VIM stimulation aligns with previous findings in cats and primates. This suggests the existence of a dedicated sensory cortical pathway that processes vestibular information related to spatial recognition and movements. This research not only sheds light on the intricate workings of our neural pathways, but also paves the way for potential advancements in clinical treatments and therapies. In a recent study published in the Journal of Neurotransmission 2024, researchers delved into the fascinating world of somatosensory Evoke Potentials SAP recordings. Typically, these recordings are done using surface electrodes. And however, in this particular study, the researchers took it a step further by utilizing deep brain electrodes that had been surgically implanted in brain targets commonly associated with the treatment of movement disorders. So what exactly are SEPs? Well, they involve stimulating a peripheral nerve, like the median nerve at the wrist, and then tracking the resulting action potential as it makes its way along neural pathways to the cortex. Along these pathways, researchers have observed time-locked potentials known as the 18 complex. These potentials are believed to originate when electrical activity reaches specific brain regions, such as the ventral intermediate nucleus ventithalamus. To conduct their study, the researchers performed evoked potential studies on patients who had different deep electrode arrays implanted in various brain targets. These targets include the subthalamic nucleus, Pedunculopontine polydicite turnus cape pallidus, and areas adjacent to the ventithalamic region. Through intracranial recordings, they made an interesting discovery. The subcortical 18 potential elicited by median nerve stimulation is generated just ventral to the VIM region. This finding suggests that stimulating the median nerve at the wrist may indirectly modulate the VIM deep brain target, potentially enhancing therapeutic effects. In fact, the researchers proposed that acupuncture point PC7 could be used as a non-invasive stimulation site to complement efforts aimed at modulating the VIM region.